All right, so get ready, because today we're diving into one of the biggest mysteries in biology: how proteins fold into those intricate 3D shapes that determine their function.
It's a field that's seen some major breakthroughs lately.
You sent over some seriously cool research on this.
Moving from simply predicting protein structure to actually designing brand new
I
ones.
know, right? It's wild. And I'm so pumped to unpack it all with you today.
Definitely. The 4 papers you shared really highlight this whole exciting trajectory.
Okay, so let's set the stage here.
Sounds good.
Proteins, they're like the tiny machines that make life tick, right?
Yeah, that's a great way to think about it.
And their structure, their shape, it dictates what they can do. Whether it's, digesting food or fighting off viruses or even making our muscles move.
That's right.
For decades, scientists have been trying to solve this puzzle,
Oh, yeah.
like how a protein's building blocks, its amino acid sequence. How does that determine its unique 3D structure?
It's like trying to solve a giant molecular jigsaw puzzle.
Totally. And the first paper we're looking at dives right into that puzzle
Okay.
with AlphaFold, this system from DeepMind that's using AI to predict protein structures
Yeah.
with incredible accuracy.
It really is amazing.
But what makes AlphaFold so different from all the previous approaches? What's the secret sauce?
Well, one key innovation is how AlphaFold approaches the problem.
Okay.
Instead of just saying, hey, these 2 amino acids are close together, it actually predicts the precise distance between them.
Oh,
So imagine you're trying to build a model from a set of instructions.
yeah.
Knowing the exact distances between the pieces would give you a much clearer picture, right?
That makes sense. It's like having a detailed map instead of just a vague idea of where things are located.
Right, exactly.
Does this distance information actually show up in the paper? Can we see it?
Absolutely.
Oh, cool. Figure 3 really brings this to life.
Okay.
They show these distance maps
Distance map.
or distograms.
Distograms, which basically depict the predicted distances between all the amino acids.
Oh, wow.
And when you compare these predictions to the actual protein structure, the match is astonishingly accurate.
So AlphaFold uses these predicted distances
Yeah.
to actually fold the protein, figure out its final 3D shape.
Right.
But how does it do that? I mean, doesn't that require tons of complicated calculations?
You might think so.
Yeah.
But that's the beauty of AlphaFold.
Okay.
It uses a relatively straightforward process called gradient descent,
Gradient descent.
guided by something called a learned energy potential.
Okay, so essentially it's like
Yeah.
the protein is trying to find its most comfortable position,
Yeah.
the one where it's most stable and energetically favorable.
That's a great way to put it.
So no need for supercomputers crunching numbers for days on end.
Exactly. AlphaFold's approach is surprisingly efficient
And elegant.
and elegant.
I like that. And this elegance, it translates into real-world results, right?
It does.
The paper shows off AlphaFold's performance in CSP13, which is basically like the Olympics of protein structure prediction.
Yeah.
CASP13, catchy name. So how did AlphaFold fare against the competition?
It blew everyone out of the water,
Wow.
especially in what they call the free modeling category.
Free modeling.
Which means predicting structures for proteins where we have no similar examples to go by.
Oh, so like venturing into completely uncharted territory.
Exactly.
That's impressive.
And even there, AlphaFold delivered remarkably accurate predictions.
Seriously. Okay, but here's where my mind is really blown.
Okay.
The paper mentions AlphaFold predicting entirely new protein folds.
I know, right?
Folds that have never been seen before.
Yeah.
Is that even possible?
It was a huge surprise, even to the researchers.
I bet.
This tells us that AlphaFold isn't just memorizing patterns from existing structures.
No?
It's actually learning the fundamental rules of protein folding.
Wow.
The underlying principles that govern how those amino acid chains twist and turn into complex shapes.
That's a game changer for understanding how proteins evolve and function.
Completely agree. It really is.
So we're talking about cracking a code that's been baffling scientists for decades.
Yep.
But wait, there's more. Paper number 2 takes things even further
Oh, yeah.
with RFdiffusion,
Right.
which actually designs novel proteins.
That's right.
We're not just predicting structures anymore.
No.
We're creating them from scratch.
Exactly.
That's incredible.
RFdiffusion uses a type of deep learning model called a generative diffusion model.
A generative diffusion model.
Think of it like starting with a blurry image and gradually adding details until you have a crisp, clear picture.
Okay.
That's essentially how RFdiffusion designs proteins.
Wow.
Starting with random noise and refining it step by step into a stable functional structure.
I love that. So RF diffusion is basically like a digital protein artist,
Yeah,
sculpting these complex molecules in cyberspace.
I like that analogy.
And what's remarkable is that
Yeah.
it can design proteins unlike anything we've ever seen in nature.
That's right.
Truly expanding the realm of what's possible.
It is.
Figure 2 from the paper showcases some of these creations.
Right.
These diverse protein monomers
Right.
that are unlike any entries in existing protein databases.
It's amazing.
That's mind-blowing.
Oh.
But are these just theoretical designs? Or can we actually make these proteins in the lab?
Well, they actually went a step further and tested some of these designs experimentally.
No way.
Yeah, so extended data figure 3 shows that these proteins actually fold and are stable in real-world conditions.
So we're not just playing with models on a computer.
No.
We're actually creating real, tangible proteins.
Yes.
With potentially groundbreaking applications.
It's huge potential.
Okay, I'm officially hooked. This is way beyond just understanding existing proteins. We're talking about creating entirely new ones, opening up a universe of possibilities.
Yeah.
But hold on.
Okay.
Before we get too far ahead of ourselves.
Sure.
The paper also mentions some limitations of previous protein design methods.
Yay.
These hallucination approaches.
Yes.
What were those all about?
So before RF diffusion, there were attempts to design proteins using methods that, well, let's just say they were prone to hallucinating.
Hallucinating, huh?
They would come up with designs that looked good on paper, but were completely unstable or just couldn't actually fold in the real world.
Ah, so like a mirage in the protein desert.
Exactly.
Looks promising from afar, but disappears when you get close.
That's a great analogy.
RF diffusion, on the other hand, is much more grounded in reality, right?
Yeah, it is.
It leverages the strengths of another protein structure prediction method, RoseTTAFold,
Right.
to guide the design process, ensuring that the generated proteins are not only novel, but also physically plausible and stable.
That's right.
So RF diffusion is like the responsible older sibling, making sure the designs are actually worth pursuing.
I like that.
It's a more reliable and efficient approach to protein design,
Definitely.
paving the way for real-world applications in fields like medicine, material science, and beyond.
And beyond.
This is all incredibly exciting. We've gone from accurately predicting protein structures to actually designing new ones.
We have.
But there's still so much more to explore.
Absolutely.
And that brings us to paper number 3, which shifts the focus to a different kind of challenge.
Okay.
Understanding how proteins interact with other molecules.
Right.
This is where AlphaFold 3 takes center stage.
Makes sense.
Okay, so we're moving beyond just the protein itself, looking at how it fits into the larger molecular landscape, right?
Yeah.
Because proteins rarely work in isolation. They're constantly interacting with other players in the cell.
That's right.
Think about enzymes, those protein catalysts that speed up biochemical reactions.
Yes.
They often need to bind to specific molecules called ligands to do their job.
Exactly.
Or consider antibodies, those immune system warriors that recognize and bind to foreign invaders.
Right.
Understanding these interactions is crucial for understanding how proteins function and how we can manipulate them for therapeutic purposes.
It's really important.
Absolutely. So how does AlphaFold 3 tackle this complex world of protein interactions? Did they just give AlphaFold 2 an upgrade and call it a day?
Not quite.
What did they do?
AlphaFold 2 was incredible at predicting individual protein structures.
It was.
But tackling interactions with other molecules, well, that's a whole different ballgame.
I can imagine.
It's like trying to predict not just how one puzzle fits together, but how it interlocks with pieces from a completely different puzzle.
Oh, okay, yeah. That sounds way more complicated.
It is.
So what did they change in AlphaFold 3 to handle this added complexity?
They made some key architectural changes to the model,
Okay.
recognizing that predicting interactions requires a different approach.
Right.
For example, AlphaFold 2 relied heavily on information from multiple sequence alignments.
MSAs?
MSAs, yeah.
Go on.
Which basically tell you how similar a protein is to other proteins in a database.
Okay.
But this information isn't always available for all the different kinds of molecules that proteins interact with.
I see. So relying on MSAs would be like trying to solve a puzzle using pieces from a different set.
Exactly.
It just wouldn't work.
Right.
So for AlphaFold 3, they had to de-emphasize the use of MSA information
That's right.
and come up with new ways to capture the relationships between different molecular entities.
Yeah.
And one of the key innovations here is the introduction of a triangle attention mechanism. Triangle attention. Now that sounds intriguing. What exactly is that all about?
Well, it's a bit technical,
Okay.
but imagine it like this. The model is now paying attention not just to pairs of amino acids, but also to triplets, forming these triangles of information.
Oh, interesting.
This allows AlphaFold 3 to learn much more nuanced relationships between different parts of a protein and the molecules it interacts with.
Okay, so it's like going from a 2D understanding of the protein to a 3D understanding of how it interacts with its surroundings.
That's a great way to put it.
Does the paper show this?
It does, yeah.
Cool.
Figure 1 tree in the paper actually illustrates this triangle attention mechanism beautifully.
Nice.
It shows how the model can capture these complex relationships.
Amazing. This is all getting pretty deep, but it's so fascinating to see how they're constantly refining these models to tackle more and more complex challenges.
It really is.
So now that AlphaFold 3 is armed with this triangle attention,
Yeah.
how does it actually perform in predicting these real-world interactions?
The results are pretty remarkable. Figure 3 shows some of AlphaFold 3's successes.
Give me some examples.
Predicting the structure of diverse complexes with impressive accuracy.
Okay.
We're talking about a ribosomal subunit with RNA and tRNA,
Okay.
an antibody-peptide complex,
Huh?
and even an enzyme with a bound inhibitor.
Wow. These are incredibly complex systems.
They are.
And AlphaFold 3 is able to predict their structures with unprecedented detail.
It really is an amazing achievement.
This is truly groundbreaking. We've gone from predicting single protein structures to unraveling entire molecular dances. But before we get too carried away celebrating,
Right.
let's not forget that even the most advanced technologies have their limitations.
Yeah, that's true.
And that's where paper number 4 comes in,
Okay.
taking a critical look at AlphaFold's accuracy
Right.
and highlighting some areas where it still needs improvement.
It's always good to step back and assess.
It is.
Even with these amazing advances,
Yeah,
it's important to remember that we're still in the early stages of this field.
that's true.
There's always more to learn.
Absolutely.
And this fourth paper dives into some of the finer points of AlphaFold's performance, especially when it comes to predicting those complex interactions we were just talking about.
Okay, sounds like we've got a lot more to unpack.
We do.
So let's dive into that in part 2 of our deep dive.
Sounds good. Welcome back.
We're just discussing AlphaFold 3's impressive ability to predict the structure of incredibly complex biomolecular systems.
It really is remarkable how much progress has been made.
It's mind-boggling to think we can now visualize these molecular dances with such precision.
I know, right?
But the fourth paper we're exploring today takes a more critical stance,
Does.
examining AlphaFold's accuracy and pointing out some of its limitations.
It's important to remember that even revolutionary tools have their boundaries.
That's true.
This paper dives deep into AlphaFold's performance,
Okay.
specifically focusing on its ability to predict interactions with ligands,
Ligands.
those small molecules that bind to proteins
Right.
and often play a crucial role in their function.
We touched upon this earlier.
We did.
Predicting ligand binding seems like a particularly tough challenge.
It is.
Why is that?
Well, ligands come in an enormous variety of shapes and sizes,
Okay.
and their interactions with proteins can be incredibly specific.
I see.
The researchers behind this paper used rigorous metrics,
Like what?
like LDDT and DOQ.
LDDT and DOQ.
Yeah, to actually quantify the quality of AlphaFold's predictions.
So what do they find? Is AlphaFold as accurate at predicting ligand interactions as it is at predicting overall protein structure?
The results are a bit nuanced.
Okay.
While AlphaFold excels at predicting the overall shape of a protein,
Uh-huh.
it still struggles with certain types of ligand interactions.
Interesting.
For example, it's not as accurate at predicting the binding of uncommon ligands,
Uncommon ligand.
those that are not frequently found in existing protein structures.
Okay. That makes sense to me.
Yeah.
If AlphaFold has primarily learned from existing structures,
Right.
it might not have enough information to accurately predict how a rare or novel ligand would bind.
Exactly.
It's like trying to solve a puzzle with pieces you've never seen before.
A great analogy.
You might be able to get the general shape,
Uh-huh.
but the finer details would be much harder to figure out.
Exactly.
Is there anything in the paper that shows this, the challenge with uncommon ligands?
Absolutely.
Oh, good.
Extended data figure 7 highlights this point.
Yeah.
It shows that AlphaFold's performance drops significantly when predicting the binding of ligands that are less common in structural databases.
Okay, so even AlphaFold has its blind spots.
It does.
But does the paper point to any other limitations beyond just uncommon ligands?
Yes.
Like what?
Another challenge they highlight is accurately modeling flexible regions within proteins.
Mm-hmm.
and disordered proteins.
Flexible regions. Disordered proteins.
Yeah,
Remind me what those are again?
sure.
Thanks.
Not all proteins are rigid, static structures.
Right.
Some have regions that are inherently flexible,
I see.
like wobbly bits that can move around.
Mm-hmm.
And then there are disordered proteins,
Yeah.
which lack a well-defined structure altogether.
Oh, right.
They're kind of like shapeshifters, constantly changing their conformation.
So they're trying to predict the shape of something that's constantly moving and changing.
Exactly.
No wonder that's difficult.
It is a major challenge for any prediction method,
Including AlphaFold.
including AlphaFold.
Right.
And they often play important roles in protein function.
They do.
So capturing their behavior is crucial for a complete understanding of how proteins work.
That makes sense.
It's a big area of ongoing research.
So it sounds like even with these incredible advances,
Yeah,
there's still plenty of room for improvement in protein structure prediction and design.
absolutely. This field is constantly evolving.
And each new discovery
Yeah,
opens up new avenues for exploration.
that's right.
The limitations identified in this paper highlight the areas where future research should focus.
They do.
This is also fascinating,
It is.
but I can't help but wonder, where does this all lead?
Good question.
What are the real-world implications of being able to predict and even design protein structures with such precision?
Now you're getting to the heart of it. The possibilities are truly vast
Tell me more.
and incredibly exciting.
I'm ready.
One of the most promising applications is in drug discovery.
Using these tools to design new drugs. That's amazing.
Traditionally, drug development has been a long and expensive process,
Yeah.
often relying on trial and error
Yeah.
with thousands of potential compounds,
Oh.
right? But with accurate protein structure prediction, we can potentially streamline this process dramatically.
So instead of blindly searching for a molecule that works,
Right.
we can actually design drugs that precisely target specific proteins involved in a disease.
Exactly.
Wow.
Imagine being able to design a drug that perfectly fits into the active site of an enzyme
Uh-huh.
that's crucial for a virus's replication, effectively shutting down its ability to spread,
Oh.
or a drug that disrupts the interaction between a protein and its signaling partner,
Okay.
halting the progression of a disease.
It's like having a molecular level scalpel,
It is.
allowing us to target diseases with incredible precision.
Incredible potential.
What other applications are there?
Beyond medicine,
Yeah.
we can use these tools to design novel materials
Material. Okay.
with specific properties.
Give me some examples.
Think about proteins that are incredibly strong,
Mm.
flexible, or even self-healing.
So we could create new types of fabrics,
Yes.
building materials,
Mhm.
or even biocompatible implants.
Exactly. The possibilities are truly limitless.
That's incredible.
And let's not forget about the potential to deepen our understanding of basic biology.
Right.
Being able to accurately predict protein structure gives us a whole new lens through which to view life's complexity.
That's a great way to put it.
We can study how proteins interact with each other in intricate networks, understand how mutations lead to disease, and even gain insights into the evolution of life itself.
It's like having a powerful new microscope that allows us to see the molecular world in unprecedented detail.
I love that analogy.
It's amazing.
It truly is a paradigm shift in our understanding of biology,
It is?
but it's also a time for thoughtful reflection.
What do you mean?
As we gain the ability to design new proteins
Uh-huh.
and manipulate biological systems at such a fundamental level, we need to carefully consider the ethical implications.
You're right.
With great power comes great responsibility.
What sort of ethical considerations are we talking about?
Well, one concern is the potential for misuse.
Misuse.
For example, in the development of bioweapons.
That's scary.
Imagine someone using these tools to design a new, highly contagious and deadly virus.
That's a chilling thought.
It is a chilling thought, but one we need to address
We do.
head on.
Absolutely.
What about unintended consequences?
Oh, yeah. Good point.
Could designing new proteins have unforeseen impacts on the environment?
Yeah. What if a designed protein escapes into the wild and disrupts an ecosystem?
That's another valid concern.
We need to be incredibly cautious
We do.
and consider the potential ecological ramifications
Absolutely.
before releasing any designed proteins into the environment.
I agree.
It seems like navigating these ethical challenges will be crucial as we move forward.
It will.
We need to strike a balance between harnessing the incredible potential of these technologies while ensuring they are used responsibly
Yeah.
and for the benefit of humanity and the planet.
I agree.
Open and honest discussions among scientists,
Yes.
ethicists, policymakers, and the public
Absolutely.
will be essential
Essential.
to guide the development and application of these powerful tools.
Couldn't have said it better myself.
Now, in part 3, I'd love to shift gears a bit
Okay.
and delve into some of the specific details from these papers
All right.
that really captured our attention.
Sounds good.
Welcome back to our deep dive, everyone. We've been exploring this incredible world of protein structure prediction and design, covering AlphaFold, RFdiffusion, and even touching on some ethical considerations. Now, for this final part, I thought it'd be fun to zoom in on some of the really fascinating details that jumped out at us from these papers. What really caught your eye?
You know, one thing I keep coming back to is the way AlphaFold actually uses those distance predictions to guide the protein folding process. It's almost like, giving the protein a set of blueprints or a map to navigate its way to that final 3D structure.
It's such a perfect analogy. And, it's amazing how it achieves this with a relatively simple process, using gradient descent.
Right.
Like, no need for those supercomputers crunching numbers for days and days.
It's elegant.
It is elegant. It's like the protein is naturally finding its most comfortable position, the one where it's most stable.
And remember those distograms we talked about earlier?
Yes, those visual representations of the predicted distances between amino acids.
It's astonishing how accurately those predictions match the actual distances in the real protein structure.
I know. Seeing those histograms really drove home the point that these aren't just like random guesses. AlphaFold's capturing something really fundamental about the protein shape.
And don't forget the fact that RFdiffusion can design entirely new proteins.
Right.
Unlike anything found in nature.
It's insane.
That says so much about the potential of these generative models.
The idea of starting with random noise and refining it, step by step, into a well-defined protein structure, just, I don't know, it's like magic to me.
And they've experimentally validated some of these designs, right?
Yeah.
Showing they actually fold and are stable in real-world conditions.
Which is incredible.
It's not just theoretical anymore. This opens up a whole new realm of possibilities for, protein engineering.
Imagine being able to design proteins with specific functions tailor-made for a particular task.
Exactly.
I mean, the potential applications in, like, biotech, medicine, material science, it's endless.
It is. Speaking of applications, the paper on AlphaFold 3 really highlighted the challenge of predicting those interactions with ligands.
Oh, yeah.
Those small molecules that bind to proteins and play a crucial role in how they work.
It's like trying to predict how a key will fit into a lock, right?
Yeah.
But there are millions of different key shapes and sizes out there, and each lock, the protein, is unique as well.
A great analogy.
So how did they even begin to approach that?
Well, the researchers had to get creative with AlphaFold 3. They de-emphasized the use of multiple sequence alignments and introduced that triangle attention mechanism.
Right.
But even then, predicting ligand binding, especially for uncommon ligands, it remains a huge challenge.
It's a good reminder that even with all these advances we've made, there's still so much we don't know.
Definitely.
But the fact that AlphaFold 3 can predict the structure of such complex biomolecular systems, it's mind-blowing.
It's a big step forward in understanding how life works at that molecular level.
This has been such an amazing deep dive.
It has.
And as these technologies continue to evolve, I have no doubt we'll see even more remarkable breakthroughs.
I agree. It's an incredibly exciting time to be following this field.
It really is.
Yeah.
So to all our listeners out there, if this has piqued your curiosity, we encourage you to dive deeper into these papers and explore the vast resources available online. There's a wealth of information out there waiting to be discovered, and who knows, maybe you'll even be inspired to contribute to this amazing field yourself.
And that's it for our deep dive into this fascinating world of protein structure prediction and design, a field brimming with potential where science fiction is becoming reality. Until next time, keep exploring, keep learning, and keep asking those big questions.
It's been a pleasure, and thank you for listening.